I and my co-authors are shown on this present slide. The computational work was done by Amir Farmani, whom you've seen earlier present on designing molecular therapy. And so this will be something completely different. This will be, we will use molecular dynamics, which accounts for the need for blue waters, but we will integrate molecular dynamics into a bioinformatics workflow, which actually makes it possible for the molecular dynamics to contribute to the solution of a real biomedical problem. And here's the biomedical problem. Viral pathogens sicken and kill millions of people each year. Mostly, we don't die from viruses. We develop antibodies that protect against viral infection, but the individual's immune response may be too little or too late to overcome the infection. And even when we develop a therapy for a particular viral strain, the strain will evolve. So we're in a continual evolutionary arms race, our immune system against all the infectious pathogens in the world. And the stakes are very, very high, and the reason Ebola has become such an important issue, an issue that captures people's attention, is that half or more of the people who contract Ebola die. As you can see from the horizontal bar graphs near the top of this graph, in Sierra Leone, 200 people died out of 550 cases, but in Guinea, far more than half died. Liberia, approximately half died. And so the human cost is enormous, and the particular symptoms of Ebola are particularly unpleasant. So when we translate this to molecular terms, here's the bottom line. Let's see now. Okay, whoops, didn't mean to do that. The surface coat proteins of the Ebola, the surface glycoproteins, are essential for recognizing surface proteins on the human cells and effecting entry into the human cells. So what your antibodies do when they are successful is they bind to the exposed part of the glycoprotein, and they block that protein from its contact with the human cell. So for a couple of different strains of Ebola, there have been crystal structures of the Ebola virus complexed with an effective antibody. For one strain, that is an antibody that grew in a mouse. In another strain, it's an antibody that's from a human being who is resistant to that strain of Ebola, who was infected, suffered from it, but his immune system kicked in strongly enough not to kill him. Now, a possible solution to this problem is to design and provide to the patient synthetic antibodies. Synthetic antibodies are becoming more and more viable. Antibodies that can be designed to be sequence specific. So one possible solution to not only the Ebola virus problem, but other viral problems, is to design and provide to the patient synthetic antibodies designed specifically against a virus strain. And the purpose of our project is to develop a protocol for computer-aided design of synthetic antibodies to speed their development and simply make it better. So here's the first proof of concept. So in molecular dynamics, we know that the molecular dynamics uses what we call semi-empirical force fields. So they're not completely accurate, they're somewhat accurate, and the question is, are they sufficiently accurate to distinguish between an antibody that will provide a good fit and therefore potentially effective therapy against a particular viral strain, or can they make that distinction? So here's the proof of concept that so if this project would have been stillborn if it hadn't passed this proof of concept. So what we did, we note that the structures of the Zaire strain of the Ebola virus glycoprotein and the strain very, very similar to each other, and the effective antibodies, the three-dimensional structure is very, very similar. So what we do is we dock the correct antibody to the correct structure and then the wrong antibody. And so what we show is that there's a significantly more structural fluctuation, and actually I could do this graph with the potential energy graph, and we'd get essentially the same result, or the mean distance that develops between the atoms in the two structures. Basically, it tells the same story. We have better energy, lower distance, and smaller fluctuations if we bind the correct antibody to the correct glycoprotein. But that's not the whole problem. That's not the whole solution. Because there are 20 different amino acids possible at each location. 20 different amino acids for each location on the glycoprotein, 20 different amino acids possible on each antibody. So the combinatorial explosion is horrible. You can't test all of the combinations, even with blue waters. So we have to now turn to bioinformatics. We have to turn to sequence-based and structure-based bioinformatics to find likely paths for the mutation of the virus and likely and antibody engineering that will likely counter the viral mutation. So the major strategies are, in the first place, we take the existing structures, we examine those structures, and we look at which residues most strongly interact with each other with respect to interaction energy and with respect to distance. So these are so when one of those residues in the virus mutates, then it's one of those corresponding residues that interact strongly with it that is likely to be an effective counter. Then we screen the possible viral mutations for statistical likelihood based on the lack of tendency to disrupt structure and the overall likelihood of substitution. And both of those, there are databases that and protocols that can do that screening, and we use the same screens for the possible antibody responses, but we add a third screen to the antibody response, and that third screen is propensity of amino acids to interact favorably in protein interiors. So that, and our hypothesis is, and this is perhaps one of the weaker parts of the hypothesis chain, that the statistics on residues that interact with each other in protein interiors are going to also interact favorably with each other at surfaces. And the only reason that that's weak is that there are different kinds of residues. That is, the residues inside proteins tend to be hydrophobic. The interactions on the surface, those amino acids tend to involve more hydrophilic or sometimes charged amino acids. So the statistics are skewed in that respect. And we can talk a little bit later about how much we think that may be influencing our results. This slide is probably not terribly visible. I apologize for that. I'm not going to go into the details of this, but I will tell you that according to our screening, there are only a few mutations that likely at each viral location that's at the interface, and there are only a few mutations that seem likely for the antibody. So we're actually going to find out that we have to expand the search somewhat above what this early assessment said. This is from a few months ago. But let's now look at some results. So let me tell you what you're looking at. The fine print is probably too fine for you to see. But on the vertical side, we're seeing the average RMSD of interacting pairs. The horizontal side, each vertical line is based on a different combination of residues. A mutation for the Ebola, mutation for the Ebola protein, and then a corresponding response for the antibody. So what you're seeing along the bottom here are a whole bunch of single viral mutations interacting that show that every single viral mutation that we make screws up the interaction. Now, here we have successfully responded. So this represents a pair where there's a mutated viral glycoprotein and we've mutated the antibody, successfully responded. Each one of these down here represents a successful redesign of the antibody to the virus. The high energy things or high fluctuation things in between represent how many antibody mutations we had to try. So you can see sometimes we hit it very quickly with 2 antibody mutations. Sometimes it took significantly longer. But most of the time we got there, and I'll show at the very end of the talk the statistics for that. This is just the output of a program called SIFT, sorting intolerable from tolerable. This is the screening step where we say, oh, there are some mutations on the that will screw up the structure, and therefore we're not even going to bother looking at those because the informatics database says they're going to mess up the structure. Here's the second screening, and this is just from an enormous array of site-directed substitutions, of evolutionary site-directed substitutions. So this database and this corresponding matrix is derived from aligning many protein families from different species and looking at individual sites where there are mutations and saying how likely are they, and then coding for the most likely ones. So that helps guide the viral mutations. And so we can rank the most probable mutations. One of the things that we notice is that the mutations that go from Zaire to Sudan, our informatics methods, we know that that's how they went, that we have the sequences. Our informatics methods, as indicated by the red structures, were mostly highly likely by the informatics. So the evolutionary path between Zaire and Sudan, viral glycoproteins, did follow a path that would be that we would rank as highly probable. So this gives us a little bit of confidence in our ability to project possible future directions. I'm going to skip through a couple of things here. This is more propensity, amino acids to be next to each other at the interface. So this is just a raw score, and this is selection based on propensity. So so finally, here's an overall picture of contact distance. Actually, the horizontal axis, I apologize, the horizontal axis got screwed up, but the horizontal axis is fluctuation size. And this is the total of approximately 1,200 simulations that were done. So the lower circle are ones that have a very good energy. So these are ones where we achieved a very, very good binding between the antibody and the viral glycoprotein. These are ones that were really terrible. And these were intermediate, and generally these were on the path from here to here. And so that'll be illustrated in the next slide. So here's a particular mutation where the first antibody response is way up here. It's got horrible distance and fluctuations, repeated attempts, and finally on the seventh trial from the antibody, seventh antibody replacement trial, we got a very, very good fit. And so presumably this particular antibody mutation would be very successful in binding to this particular Ebola viral mutation. Okay. So here, in a nutshell, I'm really restating what I said before, but just to drive it home, whoops, except that, oh no, except that the next slide I just saw is the wrong slide. Okay, we give the virus protein a relatively likely mutation, which always causes a less favorable interaction energy. Then we give the antibody a mutation judged likely to restore high affinity binding. We assay the binding affinity with molecular dynamics. We get a good restoration of binding after, on the average, 4 or 5 trials. This is longer than we expected. Now, there's an interesting question here. Why doesn't it work quite as efficiently as we expected it to work? And the answer is that every mutation, we think the answer is, every mutation causes a slight change in the structure. So every time we make a mutation, and this is the prize fight managers, you understand, the prize fighter manager always talks about but it's really the fighter who does it all. And so meaning Amir, every time a mutation is made, the structure is refined, and it's always adjusted slightly. And every time the mutation is made on the antibody, the structure is re-refined, it's energy minimized and equilibrated. And then it's the energy minimized and equilibrated structures that are docked with each other. And probably, we haven't looked in the details of this, but probably those slight changes in structure have had fairly large energetic consequences, so that the predictability of the informatics isn't quite as good as it should be. And so we have to do the molecular dynamics to keep the informatics honest. But we haven't looked into that. We haven't looked in detail at the molecular dynamics to see which particular interactions have changed and so forth, which is sort of a traditional thing to do with molecular dynamics. And now the next slide is the wrong slide. I have a slide with numbers where those blanks are. So, but that was in a different version of the show. But let me see if I can remember it. Maybe Amir can help me. So there were 99 different mutations done on the Zaire strain of the Ebola virus. there were something like 700 antibodies designed against those different 99. And 98 out of the 99 mutant Zaire strains were successfully redesigned for. So that was almost perfect success. So the efficiency was took about 7 tries, but on the average, but we got it almost every time. On the Sudan strain, there were 680 something. There were, I think, 92 mutations done on the Sudan strain. There were 600 and some odd mutations were tested to respond to them. And so far, at this writing, only 66 were successfully bound. Now, we think that with more compute time, we'll get there with the remaining Sudan strains. But that's a hypothesis to be tested. So here's the take-home message, and with my last minute or so. We integrated sequence-based and physics-based methods with an eye on contributing to the solution of an important biomedical problem. Blue water's absolutely essential, could not have been done without it due to the compute intensity of the molecular dynamics component of the project. Important distinction between here and most uses of molecular dynamics. Here we used molecular dynamics as an assay for the validity of predictions made by informatics rather than as a means to probe details of the interactions. We could go back and analyze trajectories for the details, but that wasn't our primary interest here. We believe that if the biotech industry succeeds in producing sequence-defined synthetic antibodies at high volume and reasonable cost, that the computational method we've developed can become central to antiviral therapy. And finally, I always wear this cap that says evolve, except when I'm sleeping, or a similar cap with the same message but different style, because it is absolutely central. So all of the statistics, the propensity statistics, the mutation likelihood statistics, those all were derived by comparing proteins, similar proteins from different organisms. They were done by tracking evolution. And this was essential because what we're trying to do is deal with evolution. We're trying to evolve ourselves or our technology to deal with the evolution of a virus. So I think that I have probably used my allotted time, so thank you very much.